Hello, everybody. Happy Fossil Friday. Welcome back to Fossil Friday Chats. I'm Brittany Stoneberg from the Western Science Center. With me, as always, is my co-host, Gabriel Santos.
Bom dia, everybody. Welcome to Fossil Friday Chats.
He practiced that. And welcome to today's speaker. We're very excited to introduce Dr. Paula Dentzien-Dias. Welcome, Paula.
Hi, guys. Thank you.
Hi, Paula. We're so excited to have you on the show today.
Thank you.
Yeah, we are going to learn all about coprolites, which I'm quite excited about, Paula gave us a little bit of a sneak peek before we went live, and I'm very excited for what she's going to show you, because you wouldn't think that coprolites or fossilized poop would be so futuristic looking.
It's not something I thought I would be excited to learn about when I was a kid on a Friday morning to learn about poop, but I'm really ready for you to drop some knowledge on us.
Yeah, if you guys aren't familiar with our guest today, she is a Brazilian paleontologist working mainly with vertebrate trace fossils at the Universidade Federal de Rio Grande in southern Brazil, where she teaches geology and paleontology. She's a geologist with a doctoral degree in Geosciences Paleontology. So also, if you guys have any questions for Paula, go ahead and put those in the chat. We will have our Q&A session afterwards, and you are welcome to ask any questions you might have. So, Dr. Diaz, whenever you are ready, take it away.
So you are seeing the presentation?
Not yet.
And now?
Give us one sec, folks.
We
swear we tested this beforehand.
How do you have a Fossil Friday Chats without just a few tech issues the day of?
Oh, I think that now it will go. I don't know why it wasn't working.
There we go.
There we go. Perfect.
Okay, thank you, Gabe. Thank you, Brittany, for having me here today. I'm really happy in being here with you. Well, like Brittany said, I will talk about coprolites and a little bit about inclusions and everything that we can find inside of them. But first of all, I will introduce myself for you. So, okay. So I'm from Rio Grande do Sul, Brazil. I was raised and born in Porto Alegre, and now I'm working in Rio Grande here, really south, almost Uruguay. And I am the lab head of Geology and Paleontology Laboratory and the Paleontology Curator of the Federal University of Rio Grande, FURI. Well, first, how I became a paleontologist. Well, when I was growing up, my parents used to buy to me this magazine called Super Interessante. That's like the Scientific American magazine. And I love to read everything about paleontology and archaeology too, something that I really like. And of course, I always watch documentaries about paleontology, and I love it, Jurassic Park. And when I was always playing with my dolls, my sisters didn't like it to play with me a lot, because usually in my doll house, I pretend to have an earthquake, and we are always fossil digging, me and my dolls. So that was quite tricky for my sisters to play with me. So when I grew up, I started to study geology in the Federal University of Rio Grande do Sul in Porto Alegre. And I was during my undergrad and when I was doing my master's degree, I studied dinosaur footprints. Like you could see here, a theropod footprint, and here sauropod trackway. It's not quite well preserved because it's in a dirty road, so it's a surface of a dirty road, but it's one of our best outcrops with dinosaur footprints here, because of course here in South Brazil we have all the rocks are covered by vegetation, so we usually have the outcrops are small like this. So when my I started my doctorate degree, it was when the Dr. Juan Cisneros, like you can see here, he came to the university to start his postdoc with the Permian tetrapods. And he invited me and some colleagues to work with him in the field and start to work with the Permian. And when we were doing the field trips, we start to find a lot of coprolites. In every outcrop that we went, we found one or two coprolites, always, until one day we found this outcrop that we call it coproland, that had more than 1,000 coprolites. And these coprolites are most of them from fish. So with this discovery, my study changed a lot from Jurassic to Permian, and to start the study of coprolites. But what are the coprolites? Well, they are fossil species, and they are part of this group of trace fossils called bromelites. That includes regurgitalites, gastrolites, cololites, and of course, the most abundant and more studied are the coprolites, the fossil species. Well, one thing that maybe make the researchers do not study a lot of coprolites is because sometimes it's difficult to identify them in the field. Like you can see here. Here we have some concretions that it resembles some modern feces, some animals, you know, their droppings are in pellets and small balls. So sometimes you can mistake them. And like in this example that I have here, it's a siltstone rock. And here you have one small concretion, calcite deposition. And here you have the coprolite. So if you are in the field, you can mistake and confuse with one coprolite and a concretion. So usually that makes difficult. That's something that my colleagues always say to me, oh, I never found a coprolite in the field. Probably he had found, but he had some difficulties to identify them. So when you are in the field, if you see anything that have a slightly different texture from the rock, look at that carefully, like you can see here. So here we have one coprolite, and here's the rock. If you look at carefully, you will see that it's quite it resembles a pebble, you know, like in the rock. But if you broke it, you'll see that it will have some fish scales, some fragments of bones, and the texture is completely different from the rock. Or sometimes it is already quite eroded, and you can see inside, and it will make it easier to identify them. So in my opinion, sometimes that's why usually we paleontologists say that, oh, I don't find coprolites. No, probably you found, but you have to take care of looking the rock and see these slight differences. And why we study coprolites? Well, the one thing that probably came to your mind, it's because we can reconstruct the food chain. We can know who was preyed and who ate that prey. Here we have one great paper, one work that shows that found inside a coprolite different vertebrate remains, like different fishes, like the Dipnoa, Actinopterygien remains, and even tetrapods. In one coprolite, they found a broken tooth, and they could relate that probably the producer swallowed that tooth and broke it when he was eating. So they related that coprolites to the an archosauromorph or a pterocephalian. So they could do this food chain reconstruction based in one coprolite with that information. Of course, we don't find only vertebrate remains inside the coprolites. Sometimes we can find some plant remains, like you can see here. And if you look carefully, we'll talk about it later, you can see even different things if you do, for example, thin sections of the coprolites. If you see in the microscope the coprolite, you can see cyanobacteria inside and other information that will help you to understand the environment. And that's because of this unique data that you can get from a coprolite, like cyanobacteria, as we see before. Coprolites are considered a conservatoire because they have an extraordinary fossil. And this fossil have a exceptional preservation. And it can be hair, parasites, feather, muscle tissue, different kinds of bacteria, and even insects preserved inside. So when you are studying a coprolite, you can find an infinity of information that usually we don't find in the fossil record. But how can we know who is the producer of the feces, of the coprolite? Well, the first thing that we do is look the morphology of the feces, because we know that nowadays the large carnivores, for example, they produce large cylindrical coprolites, feces, sorry. So we describe this morphology, like you can see here, the cylindrical one, but some contain even plant remains. So it's important for you to understand what's inside and the morphology. And some coprolites are unique, like this one that has a spiral in one pole of the coprolite. So that's why the morphology is so important, because it can make us understand and know how was the intestine of the animals that lived long time ago. So we know that some fishes have the spiral intestine today, and we can relate the coprolites with these groups of fishes. For example, this is scroll coprolite. That's like a papyrus, you know. Nowadays, Carchariniformes sharks have this kind of intestine. So we related when we find this scroll coprolite to this group of sharks. Or for example, the amphipolar type of coprolite, where you have the spiral along all the coprolite. It's today is produced by dipnoi, like lungfish or gars. And the heteropolar one, where you can see the spiral in one pole, it's with related to sharks, especially, and other fishes. But never forget to analyze not only the morphology, but what's inside the coprolite. So you can see plant remains inside, or bones. And this is important, and not presume if you find a coprolite, oh, because I saw this morphology of coprolite and it was related with a carnivorous animal. For example, like in this in this case, there is striated coprolites described from different periods and that have bones inside and is related to a producer that is a reptile. But this species that was found in Venezuela that has this striated surface have, like you saw in the last slide, plant remains. And that makes me think, well, I know that striated coprolites are related to reptiles. I thought, well, a reptile that eat plant, maybe a turtle produced something like that. But I started to search the morphology about the animals that live today. And we found one rodent, this one, the nutria or coipu, that is called in Portuguese ratão do banhado, something like wetland rat, in translation, that produces species with this striated morphology and with plant remains. That made us think that maybe those coprolites that are related to reptiles from Triassic, Permian, maybe they are not produced by reptiles. Maybe they are produced by a cynodont. So we have always to check the extant fauna, the extant morphology, to be sure if that morphology can be related to one group only. And never forget to analyze all the formation of that fauna, because you can compare the size and you can relate those groups with the size and what you find inside, included in the coprolite, what the animal ate with that group, like we did here with a cylindrical coprolite with plant remains. We could relate it to manatee, for example, or this cylindrical coprolite that we did not find any kind of inclusion, like bones, we didn't find. And that made us be sure that we are thinking about a producer like a crocodile, because the crocodiles have a very efficient intestine that destroys all the bones and scales, whatever the animal ate. So we were sure that we are seeing a coprolite produced by a crocodile. And of course, the one that I already told you, that was produced by a rodent. So how we search for these inclusions and everything that we can find inside the coprolite or in the surface? Why not? There's a lot of information that we can see, not only the morphology of the coprolite, but also in another kinds of information, like you can see here, invertebrate burrows. So coprophagy, not even only by invertebrates, but vertebrates that bite the coprolite or made some scratches. It can be not a vertebrate, but can be produced by a crab searching and eating on the surface of coprolites, and different kind of tooth traces, like you can see here, or even plant impressions on the surface of the coprolite. And of course, you can see bones, like you can see here, fish vertebrae and fragments of bones. And there is I think that the most common way to analyze a coprolite are the microscopical analysis in thin sections. So you cut the coprolite and make a thin slide, put in a glass, and look in the microscope. Like you can see here, a bone here, or fish scales, like these ones in the microscope, or here a plant remain, a leaf, where you can even see the cells of the leaf. Or in this amazing case where Dr. Karen Shin, found some muscle tissue inside the coprolite. Or you can find some, in the thin sections, you can find some parasite eggs, like in this case, that the parasite eggs was inside a shark coprolite, or what this means, a spiral coprolite with the heteropolar morphology. And you can see here the row of the spiral, the limits of one row you can see here, and this cluster of eggs that with the morphology of the egg and what we found inside these eggs, we can be sure that it was an egg of a tapeworm. So we know that in the Permian, tapeworms already existed, and parasite sharks in the lakes of southern Brazil. Or in the thin sections, you can find cyanobacteria, like these ones, or from the Triassic, even hair. So we know that some animals, probably cynodonts, already have hair in the Triassic. All of these way to see the coprolites are destructive because you have to cut the coprolite. So usually we only do that in the coprolites that we have a lot of specimens, because if we have only 1, 2, or 3 of that kind, we will try to do anything else but not destroy the specimen that you have. Another way to analyze the coprolite is through the scanning electron microscope, where you have to break the coprolite and cover it with some metal, usually gold, to see these structures. And you see quite good detail and everything that is inside. Like you can see here, this is a leaf, and this is very fragile structures of a leaf that you can see here, or even muscle tissue found inside the coprolite. This one was produced by an herbivorous animal, and this one by a carnivorous. So we saw this unusual preservation of muscle tissue inside a coprolite. And of course, there is a lot of bacteria and feces, and we have them in coprolites. They are preserved inside too. So here you have examples from the Permian of Russia, from the Cretaceous of United States, or even the Miocene of Venezuela. So this shows that different periods must be always studied and looked carefully, because we can find something common in coprolites, and this is one good example. And also, we found some actinomycete, these colonies of actinomycete, with the spore chains of this bacteria, the way that they produce it. and it was found inside amphipolar coprolite. You remember the one that the spiral is along all the coprolite. And here is I think that the only way to see what's inside the coprolite without destroying that example, without breaking that specimen, it's through the microtomography. And here is the amazing example that was found in the Triassic of Poland, and I will show you what they found. So you can see here with me, they found a lot of beetle fragments and a lot of complete beetles inside. So one coprolite with all of these insects, so that indicate that the producer was an insectivorous animal, and they related to a dinosaur, an insectivorous dinosaur. And they even described a new species of beetles with this amazing tomography, like you could see. And what's the most impressive thing is that the beetle is in 3D, not flattened, so you can see everything in really, really good detail. So that's an amazing new technique to look inside a coprolite without distracting them. And of course, another information that coprolites gave to us that are unique is about the environment and how the fossilization occur with these amazing specimens. So I will just tell you a little bit about another structures that we see on the outside of the coprolites. Here we have on the formation by impact of the coprolite. When the coprolite is fresh, when it's feces, and he hits the floor, it forms a surface flattened because of the way that the feces hit the floor. So you can see here. And when the coprolite is fresh, when the feces is fresh and occur a burial, a fast burial, the sediment, the weight of the sediment impact the feces and make like these lenses form of the coprolite. Or you can find some coprolites that are completely well preserved, rounded, with no kind of deformation, and that indicates that these feces had an early mineralization before the burial, before the sediment covered it. So we preserved all the structures and everything that was inside without any kind of deformation. So finally, just this talk is just a brief of about the information that we can have from coprolites, but we could see that only analyzing that the feces, we can reconstruct the trophic interactions of an ancient ecosystem. We can understand the paleo environment and understand different types of fossilization, like we saw just a few seconds ago. And we can access information about soft tissue, for example, the morphology of the intestine of animals that don't exist anymore, or even see in detail the morphology of leaves and even of muscle, and access groups that are pretty rare in the fossil record, like the parasites ones. And I would like to thank Gabe and Brittany and Heitor and Ana Emilia that helped me with this talk, and my students that helped me with some of the images. and my student that works with coprolites, with Vitória, my university, the agencies that support my research. So thank you, and I hope that my English was not that bad. Thank you. Obrigada.
Obrigada, Paula. That was so cool. I mean, I say that every week on Fossil Friday chats, but really this was like super cool, especially that 3D specimen you showed that had the beetles inside. My camera was off, so I didn't want to like you didn't have to see the stupid expressions I was making on my face, but I was just in awe watching that. Like that is so cool.
Yeah. I did not know.
That's amazing.
What was that like for you when you saw that for the first time?
Well, I reviewed that paper. Ooh. So I saw before everybody, and I was, come on, I have to find something like that too.
A little bit of professional jealousy, right? Just like, that's so cool.
Yeah. I saw some. This is the study of a doctor now. During all his PhD, he was looking the coprolites with microtomography. So all his papers was amazing, you know. Oh, new information, new ways to look inside the coprolites and find amazing things. And that one is, it's the cherry of the cake. I don't know if you use this kind of way to say that something is amazing. It's the thing that was missing in his research. I think I was clear enough to say something like this. It's amazing that paper and that finding.
I think in the past I've taken for granted like what you can learn from a coprolite, because you learn about coprolites. It's funny when you were like talking with kids like, oh, it's fossilized poop, ew, whatever. But then you showed that like you can learn so much more about an animal than from a coprolite than you ever could from its bones alone. Like I had no idea the range of morphology, or even that you can even tell what their intestine looks like based on the coprolite,
or that the preservation can be so good.
Yeah, it's just amazing.
Yeah, usually we do the technique of thin sections. This is the classic way to look for coprolites. But now with these new techniques, the SEM, seeing in detail and seeing that things that are so tiny like bacteria and different kinds of bacteria inside of coprolite. That's amazing. And we almost don't see that working with bones, that you see the total environment, not only one group or two groups, and when you have some interactions. That's really interesting.
Oh yeah, most definitely. Like I think what was really cool too is when you were talking about the way that like even the way that the fossil, the coprolite can like drop and make impressions of like leaves on the surface, that can even tell you something about like environment and things like that. That's, again, I never thought about fossils as being a Lagerstatt where there's just so much fine preservation on them. But if you think about it and take the time, it actually makes sense because of like what they are.
Yeah. You have information in the surface, inside, in one small row, like that parasite that I showed to you, the tapeworm eggs. It's inside one row of the coprolite. It's one tiny part of the coprolite. So that's why I told you before, I now I still work with dinosaurs, but my main focus is coprolite, because you have everything in one piece of poop. Mm-hmm. You have an entire environment in one piece of poop.
Well, you've definitely changed my mind on that, and I will be much more respectful to the fossil poops in our collection, and I will be much more respectful when I talk about coprolites with our students and tour groups. I'm not gonna make, well, I mean, I'll still make a joke because it's funny,
Of
but I
course.
will also remind them, like, I'll tell them, like, how cool and what you can learn from it. So, thank you.
Yeah.
Like,
Um, there's lots of great questions in the chat, so we're going to try and get to as many as we can. So thank you, everybody. Noble Cohedron would like to ask, do coprolites vary much within groups? So for example, do herbivore coprolites, like a plant eater, do they have different coprolites from carnivores, from meat eaters?
I don't think that I understand the question.
Are coprolites different between like animals that mostly eat plants and things like that, and are they different between those that eat mostly like meat?
Yeah, there's a lot of differences. It's quite rare to find some herbivorous with plants inside coprolites. And it's easy to understand why. When you eat meat and the animals eat the bones too, it's a lot of minerals that are together, and that makes easier to fossilize it. And when you eat plants, you can remember now, you can imagine a cow dropping.
Mm-hmm.
It's easy to fragment because it has a lot of plant remains, and it has a lot of food. Yeah, that's why we have the beetle dungs and a lot of animals that eat those kind of droppings. So it's easier to destroy and herbivorous poop than a carnivorous that is more dense. You understand what I'm trying to say?
Exactly. Yeah. It's interesting.
So it's rare to find those, the herbivorous ones.
Yeah. I never would have realized that would have affected the preservation in that way. That's so cool.
Yeah, there is a lot of studies that the chemistry. I didn't even talk about the chemistry because I will continue to talk.
Well, that means we have to have a part deuce of
Oh
this
my
episode.
Here's another question from Nate Carroll saying, thank you for such a great talk. Have you encountered coprolite-rich sites that also have non-coprolite phosphatic concretions?
Yeah, that one that I show you, it's a calcite. Yeah, the concretion is made of calcite. Calcite, I think right. So we have this kind of different kinds of concretion there, not phosphorus ones like the coprolites. And that one that I show you in the beginning of the talk. That's the question.
Yeah, and then more questions from Nate going into this. Let's see. What sort of taphonomic factors do you think control the accumulation? So how many coprolites are in a site?
Oh, I think that he's talking about the coprolite that I show you in the beginning that I studied. I think so. Do the question again.
Yeah, please. No worries. So what taphonomic factors do you think control the accumulation of coprolites in a site?
Well, for that particularly site, it was a pond, a lake, that we found some, how do you say that in English, drying shrinkle marks, something like that.
When the lake... yeah, I know what you're talking about.
I forgot the name in English. You know, like when the lake the water dries.
Mud cracks.
Mud cracks. Thank you. We found some mud cracks in another level, but really close to the main level of coprolites. So we think that maybe the lake dries a little bit, but not until his end. And the fishes were concentrated in one area. But the rain comes back and the level goes up again, and the fishes lives, because we almost only have coprolites. There are some in another levels with fishes and other vertebrates, but in that level only coprolites. So that's the only thing that we could imagine. Maybe we are wrong. Maybe in the future we'll think in another possibility. But until now, that's the only thing that we can imagine until now. I was clear?
Yeah, we got it.
on kind of like on that question, what causes, or like what are the environmental factors that lead to a coprolite, or a poop turning into a coprolite? Especially because you said some coprolites can be mineralized before burial. Is that correct?
Yeah. What the studies, and especially people that works with geochemistry, they say that the bacteria start in pre-mineralization thing in the coprolite. So that the fault is the bacteria. They do something. I'm not geochemistry, of course. That's not my thing. But they say that the bacteria accelerate the mineralization. So that's why we could see that kind of differences in coprolites. The ones that I showed you, that was quite one of them that's rounded perfectly and was not flattened at all. It's large one. It's like this diameter, the coprolite. So it's large one, and the weight of the sediment probably will will flatten it. And it's not flattening. And we always think that to be a good fossilization, it has to be a fast burial. It's something that at least I read and read again, and all the books say that. But with coprolites, that's not quite right. So that's why I say there's a lot of things to do with coprolites and study more, because you have a whole new world in coprolites. All the kinds of studies you can do.
Absolutely. This is, for me, this has been a great discussion about poops so early in the morning, and I think I've learned quite a bit and gained a new respect for, basically our waste.
Yeah, this is a great start.
You look it differently now.
I most certainly will.
Like, what's this? What's this?
I know this feeling.
I think you're when you're walking on the trail, and sometimes there's like horse poop or whatever. I'm gonna look at, somebody's gonna find me like on the trail, kneeling over it, looking at like, what can I learn from this? How would this fossilize? And somebody's like, are you okay? I'm like, shh, I'm sciencing right now.
Yeah, I know this feeling. When I was in New Mexico, when I met you, Gabe, I was in a trail with a friend, and I saw a bear poop, and I was crazy. My God, taking a lot of pictures, and everything was passing by me, and think, this lady is kind of crazy, but that was amazing for me. I understand, and that makes us have a lot of jokes, so that's a good thing.
Yeah, and again, it's just realizing how cool everything is. Like, everything, there's a part of it in science, and you can learn amazing things from things that otherwise you'd be like, really? That's gross. Or, we just literally flush down the toilet. It can actually learn a lot from it.
Alrighty, I think we've got one final question in the chat. So, what's the key in coprolites to tell you this is a dinosaur or this is a mammal? Like, so how do you tell the difference there?
Good question. Well, it's like I said, when you want to know the producer, you have to look a lot of things. The morphology, the size. I didn't say, but I think that's It's the information. You can understand the size, what's inside. So if there's another, for example, Karin Shin that works with dinosaur coprolites. I don't work until now. I never worked with dinosaur coprolites. She said that the morphology is not quite precise in herbivorous,
Yeah.
because if you think in a dinosaur, the large ones, until it drops and falls down, the morphology is completely destroyed.
Got a long way to go.
It don't have a quite good morphology to see like the small ones. I think that I think that he understands what I'm trying to say.
Yeah. We got it. We got that.
So,
any more questions, Britt?
I think that's it for now.
All right. Well, thank you all so much for your great questions, and thank you, Paula, for such an amazing talk. It's again, this has been a very fascinating discussion for early in the morning, but I've learned a lot, and you've really sparked a fascination, something that I want to learn more about.
Oh, thank you, Gabe. Thank you for this. It's good. It's science being shared.
Oh, absolutely. We always end the show with one final question. What piece of advice would you give to future paleontologists anywhere? And, what might you give, what advice might you give to a or someone who might be interested in looking at poop in the future?
Well, I think that what I learned during all my research is that just because you think that something it's not quite interesting, like dinosaurs. I worked with dinosaurs before, so I still work. I don't like to say I don't work anymore, but I still work. Just because something is not like that fancy like dinosaurs doesn't mean that it's not important or that you will not find an important thing inside of it. So I think that you have to be open-minded to work with science in general. So in paleontology, I think that we have to be more like this, open-minded.
Absolutely. Thank you so much for that, Paula. And then really quick, if people want to learn more about your research or want to follow you on social media, where's the best place for them to go?
Well, my lab has a page in Instagram and in Facebook, LGPFURG, if I'm not wrong. I think I can send later. So this is my lab page, and there is my personal page too, Paula Dentzien. And you can send me an email, and of course I'll answer everything that I can.
Perfect.
Yeah, fantastic. Well, be sure to put that in the chat below. And before we end, hey, Britt, do you want to put a little thing out there for something,
You might be noticing that Gabe is wearing the absolute height of paleontological fashion today. So, should you wish to visit the Western Science Center soon, you too can wear a Western Science Center t-shirt. So, go to the museum if you're in Southern California. I'm very sad that we didn't coordinate this because I could have been ready.
I was gonna surprise you. That's why I got it.
I was very pleasantly surprised. At one point, we'll have to switch, and I'll need an Alf shirt.
Okay, we'll do that. I'll send one over. Anyway, thank you all so much for joining us in today's show, and again, thank you, Paula, for such a great talk. We really appreciate you being on the show today.
Thank you very much.
As always, everybody, if you like Fossil Friday Chats and want to support programs like it at both the Alf Museum and the Western Science Center, you can find links on how to do that in the description below. And as always, make sure you like and subscribe to our channel for more stories from the world of paleontology. We'll see you all next week. Bye.
Bye.